What is stroke? Stroke is a condition that occurs when the blood supply to the brain is disrupted, resulting in parts of the body that are controlled by the damaged area of the brain not being able to function properly. According to World Health Organization, WHO, around 15 million people are affected by stroke each year. According to Southeast Asian Medical Information Center, CIMIC, Indonesia is an ASEAN country that has the highest death rate due to stroke. The Indonesian Stroke Foundation stated that the number of stroke sufferers who died in Jakarta during 2022 reached 3,600 people. And from the beginning of the year until September 2023, the number of stroke cases in community health centers reached 2,941 people. Stroke hemorrhage caused by bleeding into the brain due to rupture of blood vessels, then spreading to the brain parenchymal tissue and into space cerebrospinal around the brain. This can result in bleeding that damages brain cells and neurological functions. The brain derives most of its energy from burning glucose, consuming about 120 grams of glucose per day and producing 10 mmol of ATP, minute a gram of brain tissue. The brain needs a continuous supply of glucose as it cannot store glucose. When glucose supply decreases, the brain will switch to using ketone bodies as an energy source, such as during fasting. Disruptions to the supply of glucose and oxygen to the brain can lead to pathological conditions such as hemorrhagic stroke. When a hemorrhagic stroke occurs, rupture of blood vessels in the brain results in bleeding and damage to brain tissue. The bleeding that occurs causes an increase in intracranial pressure, disrupts cerebral blood flow and oxygen supply to a wider area, resulting in hypoxia. Hypoxia is a condition where brain tissue lacks oxygen. Without enough oxygen, brain cells begin to experience irreversible damage. Damage to brain tissue interferes with capillaries transporting oxygen, worsening hypoxic conditions, which can result in significant loss of brain function and increase the risk of long-term disability and death. This condition causes hypoxia downstream of the ruptured blood vessels. As a result, neuron cells become very active and require large amounts of energy. One approach that can be applied to overcome this condition is to increase the expression of neuroglobin, NGB. Neuroglobin, NGB, is a family of oxygen-carrying globin proteins that are found in neurons of the central and peripheral nervous systems. Mujihartini et al. 2017 reported that NGB gene expression and neuroglobin protein levels were found to be very high in the brain tissue of hemorrhagic stroke patients. In addition, hypoxic conditions in cases of hemorrhagic stroke require repair of damaged blood vessels. NGB's ability to serve as a protector of nerve cells from hypoxic damage, especially in important areas of the brain, such as the cortex, hippocampus, thalamus, hypothalamus, and cerebellum. How to increase neuroglobin? Recent research shows that NGB gene expression can be increased through therapeutic interventions, such as the use of wild plants that have potential as therapeutic agents. Apart from medical treatment, in Indonesia, there are also various kinds of herbal treatments available that utilize plants available in the surrounding environment to support the healing process. One of the wild plants that has potential as a therapeutic agent and grows widely in Indonesia is banto grass, Liercia hexandra. This grass is traditionally processed into a traditional drink called ayah banto by the Minangkabau people. People believe in the efficacy of ayah banto to eliminate toxins in the body. This shows the great potential of banto grass in the treatment of various diseases. Based on research by Anissa, 2023, banto grass extract contains various bioactive compounds such as flavonoids, alkaloids, saponins, steroids, triterpenoids, and tannins, which act as strong antioxidants. The antioxidant activity of these compounds can reduce oxidative stress in brain cells, thereby helping maintain cellular stability and neurological function. In addition, this bioactive compound is able to trigger HIF-1A activity. HIF-1 A. Hypoxia-inducible factor 1A is a transcription factor that is activated in conditions of lack of oxygen and returns conditions to normoxia through the process of regulating a number of genes. NGB expression is regulated by the HIF-1A protein, which plays an important role in cellular responses to hypoxia. Hypoxic conditions are characterized by the stability of the HIF-1 protein, which consists of the A subunit. Through the HIF-1A pathway, NGB expression can be increased, thereby helping to protect brain cells from damage caused by hemorrhage stroke. Research methods. This research is an experimental research with a completely randomized design, CRD. The tools. Materials. Research procedures. Banto grass extraction. Stage 50G. Banto grass powder was extracted by maceration using 3 solvents as much as 500 ml, which have different levels of polarity, namely methanol P.A, ethanol P.A, and distilled water. The extract obtained was then evaporated using a rotary evaporator so that the concentrated extract was obtained, and then the yield value was calculated. Phytochemical screening involves several tests for compounds such as saponins, alkaloids, flavonoids, triterpenoids, steroids, and tannins. A positive result of each test is characterized by antioxidant activity testing. The analysis of antioxidant test results is done quantitatively by looking at the IC50 value. The way is to make a linear equation curve that connects the concentration of ingredients with the percentage of inhibition. The IC50 value obtained will determine the nature of the antioxidant, following the description: very strong, strong, medium, weak. Primer design. The neuroglobin gene sequence used in this study was from Rattus norvegicus mRNA with accession number NM3 through 9 through CITSA1 The neuroglobin gene sequence obtained was stored in FASTA format for further use in the in silico primer design process using Janaeus prime. After the candidate primer pairs were obtained, the primers were selected based on the ideal primer criteria. Intermittent hypoxia treatment. This study used Rattus norvegicus rats and has obtained approval from the Research Ethics Committee of Padang State University. The experimental animals were divided into 5 treatment groups with a total sample size of 25 male rats, according to Federer's formula. These treatments are normaxia, K, HI, P1, P2, and P3. Before hypoxia induction, rats were weighed, then placed in a hypoxia chamber containing a gas mixture of O2 10% and N2 90%. Hypoxia was induced gradually with a 10-minute cycle, where 5 minutes in the hypoxic state and 5 minutes in the normal state. Brain tissue RNA isolation, stages of lung tissue RNA isolation using Genizol TM. This reagent has 5 stages, namely brain tissue homogenization, separation phase, precipitation phase, RNA washing, RNA resuspension, calculating RNA concentration and purity, cDNA synthesis using the Sensife STM cDNA synthesis kit, BIO 6053. Total RNA was homogenized using a vortex. After that, it was incubated using a PCR machine with primer annealing steps at 25 slice for 10 minutes, then reverse transcription at 42 slice for 15 minutes, and inactivation at 85 slice for 5 minutes. cDNA was temporarily stored at 4 slice. Gradient PCR is a step for optimizing the primer annealing temperature before being used for gene expression analysis using QRT-PCR. PCR according to the MyTac HS Red Mix BioLine Instruction Manual. The annealing temperature was gradient in the range 57 to 58.7 for 15 seconds. PCR gradient products were analyzed by electrophoresis using a 1.5% agarose gel, 100 V for 27 minutes. Quantitative real-time PCR. The QRT-PCR reaction components are mixed according to the composition determined from the 2X Sensifast SYBR No Kit. ROX with details of initial denaturation reaction at 95 degrees for 1 minute, denaturation cycle at 95 degrees for 15 seconds, annealing at 60 degrees for 15 seconds, elongation at 70 degrees for 10 seconds, set cycle 35 times, final elongation at 70 degrees for 5 minutes. Data analysis research data was analyzed quantitatively. NGB gene expression was analyzed by relative quantification using the Leavik method. Then statistical analysis was carried out using SPSS via the ANOVA test at the 5% level. Result: evaluation of phytochemicals and antioxidants in Banto grass. The results of phytochemical screening of Banto grass extracts with 3 types of solvents, methanol, ethanol, and distilled water, showed that Banto grass contains saponins, flavonoids, alkaloids, steroids, triterpenoids, and tannins. The following are the results of the identification of phytochemical content and antioxidant activity of Banto grass extract. Description: use contains compounds, does not contain compounds. The antioxidant activity of Banto grass with methanol extract is very strong at IC Phyt 39.35 RL. Primary design results. This stage consists of several analyses, starting with designing the neuroglobin primer as a sequence that can amplify the target nucleotide sequence, testing the effectiveness of the BLAST primer, and optimizing the NGB gene primer. Genius Prime software generated 2 pairs of candidate primers to amplify the NGB gene mRNA in the 920-1722 region of Radus norvegicus. The 2 pairs of primers designed by Primer BLAST showed variability in terms of PCR product size, primer length, TM, and GC percentage. The following are the results of the primer design results from Primer BLAST. The product produced by a pair of primers is 803 BP. Furthermore, the melting temperature, TM, of the primer pair is 57.10 C and 58.0 C. Based on the table, the results of the primer design stage obtained 1 advanced primer for neuroglobin Radus norvegicus, NGB, which has a length of 20 bases, with the sequence 5 TOS AGTCTTGCCCTCACAG, thruses, and 1 reverse primer with a length of 20 bases, with a sequence 5C GTC ACAG AACACGGACA 3 EU, product size 803 BP. The TM difference in this primer pair is 0.9 degrees C. The design results meet the good criteria so that the designed candidate primers can be used for the PCR process. Optimization and effectiveness of NGB genes. The synthesized primer pairs were optimized using a PCR gradient to obtain the appropriate annealing temperature. The following are the results of the PCR gradient test using NGBF and NGBR primers: A 57 GB, 57.7 G, 58.2 on ZD, 58.7. Based on the results in the figure, the optimal temperature for NGB forward and NGB reverse is 57.7 Z as shown in letter B by showing the thickest and clearest DNA band. This indicates that the primer can attach to a DNA template region at 57.7 degrees C, which is complementary to the base sequence. Analysis of relative expression of NGB gene mRNA in rat brain by qRT-PCR. Analysis of the relative expression of the neuroglobin gene was carried out using the Leavik method. The data used is the average CT value for each treatment sample for the neuroglobin and 18 RNA genes. The results of the analysis of the relative expression ratio of the neuroglobin gene in the brains of mice from various groups are shown in the figure. Average relative expression of neuroglobin genes in mouse brain. Graphs followed by the same letter notation indicate treatments that are not significantly different. ANOVA P00.05. The graph shows the results that the highest relative expression ratio of the neuroglobin gene was in the first treatment group with a value of 21.10, while the lowest relative expression ratio of neuroglobin was in the normoxia group with a value of 1000. The relative expression ratio of the neuroglobin gene in treatment groups 2 and 3 decreased compared to treatment group 1. This means that administration of bantograpss extract can maintain optimal neuroglobin gene expression in treatment group 1 at a dose of 50 mg BW day. Potential results from this research include NGB primers that have been designed, and potential results from this research include NGB primers that have been designed and optimization based on the mRNA sequence Rattus norvegicus neuroglobin, NGB, with NCBI accession number REF NM03359.3, can be used to amplify the neuroglobin gene and has obtained an IPR copyright certificate. This means that the use of bantograpss extract makes it possible to maintain and modulate the presence of oxygen through NGB expression and becomes a new drug candidate to prevent hypoxia in cases of hemorrhage stroke syndrome. Conclusions. Based on research activities that have been carried out, it can be concluded that administration of bantograpss extract, L-hexandra-SW, had a significant effect on the relative expression ratio of the NGB gene with the highest value found in the first treatment group, namely 21.10, and the lowest ratio was in treatment groups 2 and 3 with the provision of 1,000 bantograpss. The dose of bantograpss extract, L-hexandra-SW, which can influence intermittent hypoxia through the expression of the neuroglobin, NGB's gene in the rat brain, is 50 mg BW day. The administration of bantograpss extract can act as an additional antioxidant that restores intermittent hypoxia to normal.